Hello everyone, welcome to our YouTube channel. Today in this video, we are going to see basic fundamentals of affinity chromatography. So without taking your too much of time, let's get started. There are so many blogs available on Google, but not as specific and detailed. Here I am trying to solve this problem and give a complete and detailed note on affinity chromatography. In this in-depth video, you will learn first, what is affinity chromatography, who discovered affinity chromatography, what is the principle of affinity chromatography, how does affinity chromatography work, what are advantages and disadvantages of affinity chromatography, and what are applications of affinity chromatography. So if you are ready to go all in with this video, let's dive right in. First, what is affinity chromatography? Affinity chromatography is a type of chromatography which is a separation technique. It is also called as a sample purification technique. This technique is basically used for biological molecules, for example various proteins. By means of specific interactions of molecules, this method separates our desired molecules from the mixture. This specific interaction is done with the component known as a ligand. In this technique, the ligand is immobilized on a support. To understand the basic working process of this technique, suppose we are passing a solution of a mixture of proteins through a column. The separation will be done on the basis of affinity. If the proteins have a higher affinity with the ligand on the column, it will stay there for a longer period of time and other molecule will elute faster. Likewise, the separation takes time on the basis of affinity. Here, ligand and molecules interact just like a lock and key model. So, let me summarize the definition. Affinity chromatography is a separation method that involves the separation of biochemical mixtures based on the highly specific interaction between enzyme and substrate, receptor and ligand, or protein and nucleic acid, etc. Who discovered affinity chromatography? In 1987, Pedro Quatrecases and Meir Wilczek discovered and developed affinity chromatography and its applications to biomedical sciences. What is the principle of affinity chromatography? It is a separation technique which involves the separation of desired product from the mixture on the basis of affinity. How does affinity chromatography work? Actually, there are various kinds of procedures seen in USP, that is United States Pharmacopoeia, BP, British Pharmacopoeia, or IP, Indian Pharmacopoeia, but all follow the same concept in the background. You can follow this procedure for your lab work. Here the matrix which is on the column is in bead form, to which a specific ligand is bound by a covalent bond. In order to be effective for a matrix, it must have certain parameters. For example, it must be insoluble in buffers and solvents employed in the process. It must be mechanically and chemically stable. It must be easily bound to a ligand or spacer arm onto which the ligand can be attached. It must show good flow properties and must have a relatively large surface area for the attachment. If an enzyme is to be purified, a substrate analog, inhibitor, cofactor or effector may be used as an immobilized ligand. Attachment of ligand to the matrix. First one is preparation of column. Initially, the column is loaded with solid support such as cellulose, saccharose, agarose, etc. Ligand is selected based on the desired isolate, and spacer arm is attached between the ligand and solid support. Second, sample loading. A solution having a mixture of substances is poured into the elution column and allowed to run at a controlled rate. Third, ligand molecule complex elution. The target substance is recovered by changing the conditions to favor the elution of the bound molecules. Now, here is the procedure that you can follow. Firstly, prepare your column part. For the column part, first take the inner material of the column, which is called a stationary phase, which is a cellulose, saccharose or agarose. These all are basically used as a stationary phase because they are easily available and comparatively cheaper also. Now you have prepared the column by putting the stationary phase inside the column, and then you should fix it on the stand. Then the ligand and spacer arm is added to make a solid matrix, which will hold our solution mixture for a certain period of time for the effective interaction with the ligand and stationary phase. Then the sample loading is to be done, in which we have to load our sample from the above side of the column, and then the solution mixer will react with stationary phase for some period of time. After the completion of the interaction, our desired material is eluted from the column, which indicates the completion of separation process. Now you can use your sample for further analysis. Here the process of separation is completed. Now we are going to discuss commercially used gels that are widely used and easily available. List of commercially used gels. First, Cyanogen Bromide activated Agarose. Second, 6-amino hexanoic acid Agarose and 1,6-diamino hexane Agarose. Third, epoxy activated Agarose. Cyanogen Bromide, that is activated Agarose. This gel is used widely because it is available and comparatively it is compatible with almost all materials. All ligands which are containing primary amino groups are easily attached to the agarose, and that is why this gel is especially versatile. Very gentle conditions may be used to couple the ligand since the gel is extremely reactive. Second, 6-amino hexanoic acid agarose and 1,6-diamino hexane agarose. Between ligand and matrix, steric interference problems are seen, and these activated gels help overcome the steric interference problem by placing a 6-carbon spacer arm between ligand and matrix. Ligands containing pre-primary amino groups couple covalently with CH agarose. Ligands containing pre-carboxyl group couple covalently with AH agarose, that you have to keep in mind. Third, epoxy activated agarose, which is used for the attachment of ligands containing thiol, amino or hydroxyl groups. Fourth, group specific adsorbents. Vibration blue agarose specifically reacts with enzymes which are having nucleotide cofactors like DNA polymerase, serum albumin and kinase. Now let me show you the components of affinity chromatography. First one is matrix, second is spacer arm, and third one is ligand. Matrix. In order to be effective, it must have a certain characters. It should be insoluble in buffers and solvents employed in the process. The matrix is actually inert support to which a ligand can be indirectly or directly joined. The matrix should be inert physically as well as chemically and must be easily coupled to a spacer arm or ligand onto which the ligand can be attached. It must show good flow properties and should have a considerably large surface area for effective attachment. It must be chemically and mechanically stable. Mostly used matrix materials are polyacrylamide and agarose. Second one is spacer arm. Spacer arm is used to improve the binding between the target molecule and the ligand by removing any effects of steric hindrance. Third, ligand. The molecule that binds reversibly to a specific target molecule is called as a ligand. When we know the nature of the macromolecule to be isolated, only then we can select the ligand of our choice or requirement. The hormone itself is a good or an ideal candidate for the ligand when our hormone receptor protein is to be purified by using this chromatography method. For the isolation of antibodies, a heptane or antigen may be used as a ligand. Now let me show you the limitations. Firstly, it consumes a large amount of time. When the solution mixer comes at the bottom of the column, it needs some time to react with the support or stationary phase for the better separation, and that is why it takes time. As the columns contains affinity resins and other stuffs in compacted form, that is why the solution mixer takes time to come at the bottom from the top. It is expensive because more amounts of solvents are required. Non-specific adsorption cannot be removed, it can just be minimized. In this chromatography method, there is limited availability of immobilized ligands along with their high cost. If the required pH is not adjusted, proteins get denatured. You know very well that everything which has its application is preferred first in the market. So now let me show you the application of affinity chromatography. First, it is one of the most useful methods for the separation and purification of specific products from the mixture. Second, it is a sample purification technique and it is used basically for biological molecules, for example proteins, nucleic acid and enzymes, etc. Third, it has its application for concentrating and purifying the compound from a mixture. Its major application includes: first, it is used for the separation of a mixture of various compounds. Second, it is also used in the purification process or removal of impurities. Third, in the assays of enzyme. Fourth, substrates detection. Fifth, for the investigation of enzymes binding site. Sixth, it is used for in vitro antigen antibody reactions. And last, it is also used for the detection of single nucleotide polymorphisms and knowing the mutations in nucleic acids. What are the advantages and disadvantages of affinity chromatography? Advantages. First, mostly it is used to study enzymes and proteins in the pharmaceutical field. Second, in the pharmacy world, affinity chromatography is used to check the quality of the proteins and the enzymes which is present in the formulation. Third, it's also used in genetic engineering field for the gene therapy and identifications of genes. Fourth, it is also used during the production of vaccines. Fifth, this chromatography is mostly used in pharmaceutical company in the production department of the vaccines for maintaining the quality of the product. Sixth, in pharmacy and other research work where they require high purity, it can be achieved by affinity chromatography. Seventh, high specificity is achieved with affinity chromatography. 8th, for increasing the solubility. 9th, it prevents proteolysis. 10th, the elution column is related to molecular weight. 11th, in affinity chromatography there is less zonal spreading compared to other techniques. 12th, it does not depend on the temperature, pH, ionic strength and buffers composition. Therefore, separation can be carried out under any condition. 13th, high degrees of purity can be obtained. 14th, the binding sites of biological molecules can be simply investigated. 15th, in affinity chromatography the process is reproducible. It is major advantage. 16th, highly sensitive for the detection of compound like halogen, peroxides, quinines, nitrites, etc. 17th, it is more sensitive than TCD and FID. Now let me show the disadvantages of affinity chromatography. First, compared to another chromatography method, the affinity chromatography is non-specific for adsorptions. Second, ligands used in this method are too much expensive, that is why it is used in specific work only. Third, it has low productivity. Fourth, sometimes ligands leakage problems are also seen. Fifth, the presence of metal ions contaminates the purified protein solution because they may destabilize or stabilize the protein. Sixth, metal ion transfer and metal ion leakage lead to protein loss. Seventh, degradation of the solid support. Eighth, expensive ligands. Ninth, interfere with structure. 10th, limited sample volume. 11th, for using it, the person must have skills to handle it. And 12th, considerable optimization is required.